This talk is intended for medical students and residents starting their neurology rotation. This talk is about the organization of the nervous system. The nervous system is divided in the central nervous system, which includes the brain and the spine. The second division of the nervous system is the peripheral nervous system, which includes roots, plexus, nerves, and ganglia. I will now represent the two main divisions of the central nervous system, the brain and the spinal cord, to talk about their subdivisions. The brain can further be divided into three subdivisions, as you can see in this diagram. These divisions are the cerebrum, the brainstem, and the cerebellum. The cerebrum can be subdivided into three parts: the cerebral hemispheres, the basal ganglia, and the diencephalon. A good view to start looking at the cerebral hemisphere is from the front. From the front, we can identify the longitudinal fissure, the lateral fissure or sulcus, many less deep sulci, and many convolutions. But to really see what the cerebrum is made of, we must look inside. I have introduced a lateral view of the brain to show the site of cut that will yield this view. In this view, we can see enclosed in aqua the cerebral hemispheres, the longitudinal fissure, and the lateral fissure. The cerebral cortex, made of gray matter forming a mantle, convolutions, and sulci. But in addition, we can see white matter. The next part of the brain we will briefly talk about is the basal ganglia. Here you can see the same brain cut that you were seeing a few seconds ago. And enclosed by the magenta line, you can see masses of gray matter. These masses are called basal ganglia. The main components of the basal ganglia are the putamen, which means the shell, because someone thought they looked like shells, the globus pallidus, which means the pale ball, and the head of the caudate, which means the one with the tail. And indeed, as you can see in this representation, the caudate has a tail. After these few words about the basal ganglia, I'd like to talk to you about the last division of the cerebrum, the diencephalon. Going back to the anterior brain view, cutting through the middle right down through the longitudinal cerebral fissure, and then disregarding the left hemisphere, as I have done here, and rotating the hemisphere as shown, we would display the medial surface of the brain. This way we can see the diencephalon, which is the area I have enclosed in magenta. The diencephalon has 4 parts, but in this view we can only see 3 of them. The thalamus, the relay station for the cerebral cortex, the hypothalamus, the center that controls autonomic and endocrine functions, and the epithalamus, which has an endocrine function. The 4th component of the diencephalon, the one we cannot see in this view, is called the subthalamus. To see the subthalamus, we must go back to the anterior view of the brain, but this time include in the specimen the upper part of the brainstem and cut not anteriorly as we did before to look at the basoganglia, but more posteriorly to obtain this view. In it, you can see the thalamus, also called the dorsal thalamus, the red nucleus, and the substantia nigra. We must now use a neural stain to impregnate the tissue. As you can see in this frame, to be able to see 4 less well delineated structures than the 3 we have just mentioned. These structures are the zona incerta, an important nucleus in the control of movements, the nucleus of the field of Forel, also involved in movement coordination, the entopeduncular nucleus, another movement coordination area, and the subthalamic nucleus of Lewis. These 4 nuclei, the rostral aspect of the red nucleus and the rostral aspect of the substantia nigra, constitute the subthalamus. That is also called the ventral thalamus. Why is this area important? Because these nuclei are involved in a rare and dramatic disorder characterized by violent movements called hemibalism, usually due to a stroke in the subthalamic nucleus, and a very common and devastating disorder called Parkinson's disease, due to degeneration of the dopa-secreting cells in the substantia nigra. Now that we have looked at the cerebrum components, we will look at the brainstem. The brainstem has 3 components: midbrain, pons, and medulla. We will talk about these structures later on. The last division of the brain is the cerebellum. Again, we will talk about it in a future talk. About the spine, I would like to tell you now that it is a cylindrical tube with no anatomical segmentations, but with visual segmentations dictated by the specific area of the spinal cord that contributes to a single spinal nerve. So we talk about 8 cervical spinal segments because there are 8 cervical spinal nerves. The fact that in the spinal column there are only 7 cervical vertebrae dictates that the first spinal nerve exits the spinal canal between the skull and the first vertebra, as you can see in this frame. and that the C8 spinal nerve leaves the spinal canal below the 7th vertebra. In addition to 8 cervical segments, we have 12 thoracic segments, 5 lumbar segments, 5 sacral segments, and 1 or 2 coccygeal segments. The lower part of the spinal cord, where the size of the spinal cord tapers, is called the conus medullaris. The tip of the conus medullaris does not go all the way down to the bottom of the spinal canal. In adults, the tip of the conus ends at about the lower level of L1 vertebra or the upper level of L2 vertebra. Thus, the nerve arising from the conus medullaris and for some distance above it travel in the vertebral canal for some distance to reach their corresponding intervertebral foramina. This structure is called the cauda equina. This is the end of the section on the overall organization of the nervous system. Thank you.